Welcome to OA Week 2023. I hope you've all been able to join. I would like to welcome you and thank you for being here. My name is Katharina Schuh. I'm with the Intergovernmental Oceanographic Commission of UNESCO. And I would just like to point out that this session is being recorded. So OA Week is sponsored by 4 organizations. First of all, GOA-ON, the Global Ocean Acidification Observing Network. Secondly, NOAA, the United States National Oceanic and Atmospheric Administration. And third, the IAEA OAICC, the International Atomic Energy Agency, Ocean Acidification International Coordination Center. And last but not least, the IOC of UNESCO, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization. So GOA-ON was established in 2012 with just a handful of members. Since then, GOA-ON has grown immensely, as you can see on this map. We now have over 1,000 members from 114 countries. GOA-ON also includes 9 regional hubs, which span continents and oceanographic regions. We'll be hearing from most of these regional hubs throughout this OA week. If you're not a member yet, you can join GOA-ON by visiting GOAON.org. OA Week debuted in 2020 and returned in 2021, when events and conferences were postponed due to COVID-19. Following the successful in-person symposium on the ocean in a high CO2 world in 2022 in Lima, Peru, GOA-ON is bringing back OA Week this year in 2023 to maintain momentum around OA research and to provide a virtual platform for the ocean acidification community to exchange their latest findings. We're thrilled to present a wide range of ocean acidification topics and speakers from around the world. During the presentations, please note that all participants are in listen-only mode. You're welcome to type any questions into the questions box, which can be found at the bottom of the control panel on the right-hand side of your screen. We will be monitoring the incoming questions and will pose them to our speakers following their presentations. For the discussion, you can also use the raise your hand function in the toolbox at the bottom of your screen, and we will call on you to ask your question directly. And with this, I would like to introduce to you the moderator of this session. This is Sylvain Agostini. Sylvain, if you could join us.
Hello, everyone.
Good morning. Sylvain is with the University of Tsukuba and the ICONA network. And Dr. Agostini is studying the effects of climate change and ocean acidification on the ecophysiology of corals, focusing on coral ecosystem in marginal areas. He studies the response of marine organisms and communities exposed to multiple stressors, taking advantage of natural analogues and gradients in CO2, latitudinal temperature gradients, and similar. He initiated the use of the Shigeki Island CO2 sea and is a co-founder of the ICONA network, which he'll be telling us more about now. Over to you, Sylvain.
Thank you for the introduction. So I think we'll go straight into the different talks. So today we'll have talks from ICONA members, including Shigeki Wada, as first Dr. Carlo Cattano, and then 2 early career scientists, Ayaka Umeda-san and Leila Ijima-san. So, well, the ICONA network is a network that was created to foster the use of natural analogues, including silt seeps and natural anthros bay, that can teach us a lot about the ocean acidification effect at the ecosystem level and also the chronic long-term effect. But without losing any more time, I would like to introduce our first speaker. So our first speaker is Dr. Wada Shigeki from the University of Tsukuba, who is working on biogeochemical cycles and especially the importance of microalgae on carbon cycle for the carbon cycle. And recently he has been working a lot on the ocean acidification effects on carbon cycle in coastal water, and including the use of natural analogues, and especially the CO2 seep of Shigeki Island. So Wada-san, if you want to start your presentation, the floor is to you.
Okay, thank you. Trying to share. So really wait.
In the meantime, so as Katerina said, for the questions, we'll take questions at the end of each presentation and hopefully also at the end of the full panel.
Okay, can you see?
Yes, it's fine.
Okay. Okay, thank you, Sylvain. I'm Shigeki Wada from Shimoda Marine Research Center, University of Tsukuba. So today I'd like to talk about the community photosynthesis in coastal ecosystem and the high CO2 world. So as you know, the oceanification is one of the most serious problems of marine ecosystems. So this figure shows the time course of the pH in the western North Pacific. So, and from the 1983 to 2008, so for 25 years, so pH continued to decline. And so oceanification has various effect on the marine organisms. So some organisms has a positive effect and some organisms negative effect. So for example, about the seagrass, but it depends on the species, but seagrass is generally considered to be positively impacted because they prefer the high CO2 conditions. But so calcifying organism is negatively impacted. And also, for example, the snails, the shells, corals, etc. So they, in such a calcification, is strongly suppressed by the oceanification. So the effect of the oceanification is species specific. So it's quite difficult problem to predict the marine ecosystems. So so this session is the ICONA, and so we made the ICONA network. So ICONA is the International CO2 Natural Analogue Networks, and so we use the natural analog to predict the effect of the oceanification on the marine ecosystems. And we have the two approaches, two types of the study sites. So one is a volcanic CO2 vent, and today's my talk is focused on this. And so around the volcanic CO2 vent, the CO2 gas is released from the sea floor, and CO2 is dissolved into the waters. So, the ecosystem around the volcanic ecosystem around the vent is naturally acidified. So please look at this map. So this map shows the distribution of the PCO2 concentration, and so this is Shikine. So in this bay, there is a volcanic vent, and red color shows the high CO2 concentration. So we usually use this area as to predict the future system. And so please look at this area. So this area is also mostly blue colors. Blue color shows the CO2 level at the present ocean. And so we usually use this area as a reference site. So another type is the semi-enclosed bay, and maybe the other speaker will talk about that. So today's my talk is just focus on this. And about the volcanic CO2 vent, we use the several sites for the oceanification research. So we are in Japan, the Shikine Island is one of the sites of the CO2 ship. So in the reference site, this area, the many coral and canopy forming algae, but in the high CO2 side, the coral is disappeared, and the body size of the macro is very small, so low profile or tough algae dominates. And we also using this network, we also sometimes we visit other areas, and we can compare the ecosystem because there's a various different ecosystem in its seeds. So anyway, so my talk is focused on macroalgae and photosynthesis. So macroalgae is one of the foundation species in coastal ecosystem, and so there's a various types of the macroalgae, and so they produce the organic matter and energy by the photosynthesis, and they support the energy flow and material cycle and carbon cycle, and sometimes so energy is transferred to the higher trophic level. So the photosynthesis is one of the most important driver of the ecosystems, process of the ecosystems. But photosynthesis is one of the thought is the photosynthesis is affected by the ocean acidification. And so, for example, in the photosynthesis process, one enzyme, there is one important enzyme. It is rubisco. To capture the carbon dioxide and convert to the organic matter. So this enzyme is quite important to catch the carbon dioxide. But the performance of this enzyme is not so great at the low CO2 levels. So for example, the CO2 level at the present ocean is around 10 to the 15 micromolar, but half saturation concentration of this enzyme is 20 to 40 micromolar. So the present CO2 level is quite lower than half saturation levels. So the increase in the CO2 level will be double or triple in the future, and so it is close or sometimes over the half saturation concentration. So activity of the rubisco will be enhanced by the high CO2 condition. So if we simply assume the activity of the rubisco, photosynthesis will be enhanced by the high CO2 condition. But there is some complicated process. So one of the important process is the CCM. CCM is a CO2 concentrating mechanism. And so the rubisco is localized in the chloroplast. And if the algae is actively transported and localize the CO2 around the rubisco enzyme, so the performance of the rubisco will be greatly enhanced. So at the situation, the simple situation is this is a rubisco and low concentration of the carbon dioxide around the rubisco. But in the seawater, much amount of the bicarbonate ion is present. And if the algae can localize and concentrate the carbon dioxide around the rubisco, it will enhance the activity of the synthesis. So, and one of the process of the CCM is the conversion of the carbon dioxide from the bicarbonate ion. And in this process, carbonic anhydrase is affected, and they convert the bicarbonate into the carbon dioxide. So, and the metabolic process is, if we consider the metabolic process, the prediction of the photosynthesis is very, very complicated. And another important factor is the change in the flora. So in the normal, in the present level CO2, some flora is living, but so in the high CO2 condition, the flora will change because of the effect of the ocean acidification. So this is an example in the Shigeki Island. So in the 300 ppm, the reference site, there is a coral and the canopy forming algae. So body size of the algae are large. But in the high CO2 site, the coral is disappeared and low profile algae and the tough algae are dominant. So, and so the photosynthetic activity is different among the species. So considering such a difference, it's also the factor. It is so considering the response to metabolism and the change in the flora, it is difficult to predict the community production in the future. So our objective in this study is to estimate the community photosynthesis using the natural analog of photosynthesis. And here I would like to introduce the results of the two approaches. And one is the deployment of the chamber. So we use the transparent chamber and deploy on the sea floor and directly measure the photosynthesis. And another approach is the consideration of the movement of the water mass. So in this approach, we deploy the oxygen sensor and current meter. And so the water mass is moving with the water current. And for example, that's from A to D, the water mass is moving. And if the photosynthesis, the macroalgae produce the oxygen because of the photosynthesis, they supply the oxygen. So the oxygen concentration at A is lower than B. So using such a difference of the oxygen concentration, we can calculate the community photosynthesis. Okay, first I would like to move to the first approach to the community photosynthesis. So our study site is the Shigeki Island, and we have the and so there is a volcanic CO2 band, and this area is a high CO2 site, and this area is a reference. So the seascape is completely different. So in the reference site, there's a coral and the large algae, but in the high CO2 site, the sea floor is mostly covered by the low profile or tough algae. So here I'd like to show you the community composition. So we categorize the season into the pre, post, and recovery from typhoon because typhoon is important factor determines the benthic community according to our previous studies. So, and the R and H shows the reference on the high CO2 site, and please compare the R and H. And in post and pre and recovery season, each season, in the reference site, the coralline algae and branched coralline algae and canopy forming algae are dominant. But in the high CO2 site, the brown color, it is a low profile algae. And sometimes the tough algae, it's a pink color, it's there appeared. So these are low, but small body size or algae are dominant in the high CO2 site. And this is the community results of the community photosynthesis. And horizontal axis is light, and vertical axis is net photosynthesis. And so our approach is carried out in situ, so we cannot control the light intensity. But light intensity is an important factor to determine the net photosynthesis. So here we draw the correlation line between the light and the net photosynthesis. And our hypothesis is the high CO2 fertilize community photosynthesis. So if it is true, the slope of the light versus net photosynthesis will be higher in the high CO2 site. And So R, so please compare the slope value, and R is in the post-season is 0.62, and high CO2 site 0.86. And pre-season is 1.3, 1.3 is very, very similar. And the recovery season is high value is the reference site. So based on the linear mix model shows no significant difference between the two sites. So based on this data set, we cannot show the any CO2 fertilization regardless the difference of the CO2 levels. So, and we also did another approach using the deployment of the sensors. So the method of this approach, this approach is also carried out in the Shigeki Island and the mostly same location in the high CO2 and the reference site. And in this approach, we deploy the oxygen sensor. So for example, blue color, the location of the blue color is the site deployed with the oxygen sensor. And so red color also is the position of the oxygen sensor. And black color is the location of the current meter and the photosensors. And so here I'd like to show you the example of the result, raw data. So in this result, the yellow color shows the northward current. So, and so 0 is the middle. So if the value is so there's a negative value, it shows the southward, the direction, the current is the southward direction. So in this case, the station 1 is the downstream side, and upstream side is station 3. And please look at the dashed and solid line. And dashed line is oxygen concentration at the station 3, and solid line is the station 1. And there's a difference in the this period. So, so in this case, the higher oxygen concentration is found in the downstream side. So it would reflect the production of the oxygen in the area between these two sites. And using the difference of the oxygen concentration, we calculate the photosynthesis. And so difference of the oxygen is evaluated by the DO sensor, and time of the movement of the water mass from the station 3 to station 1 is calculated by the current meter. And using this equation, we can calculate the net photosynthesis. So this is the result, overall the results for this approach. And the vertical axis net photosynthesis, and horizontal axis the light intensity. And reference site and high CO2 site is mostly similar levels. So we draw the correlation line, and the slope is the reference site 3.4, and the high CO2 site is 3.3. So mostly similar slope values implies no CO2 fertilization between the sites. So here I'd like to discuss why there's no difference of the synthesis. So, sorry, and so if we simply consider the activity of response of the rubies against the CO2 levels, the photosynthesis will be enhanced by the increase in the CO2 levels. However, our results shows no change of the net photosynthesis under high CO2 condition. So we thought the level of the photosynthesis at the present ocean has been already enhanced by the adaptation to the low CO2 level condition. So here we consider the effect of the CCM. So as I explained before, the CCM is concentrating CO2 to the high. So it's a mechanism to supply the high concentration of the CO2 to this. And one of the important the process is the conversion of the bicarbonate ion to the carbon dioxide with a carbon anhydrase. And between these two molecules, there is an isotope discrimination. So carbon-13 value of the bicarbonate ion higher than carbon dioxide. So we can distinguish the carbon source based on the stable isotope value. This approach has already done by the other researchers using the CO2 ship in the Vulcan Island. So they show the change in the community composition, and the dark bar is the CCM user, and at the normal pH level, CCM user is dominant, but in the high CO2 side, CCM user declined. So the carbon-13 value is the indicate of the efficiency of the CCM process. And we also measure the carbon-13 value, and our approach is the community level assessment. So we also calculate the community average the carbon-13 value. So to calculate this, we use the carbon-13, the data of the carbon-13 of each species and coverage of each species. So we multiply these values, and some of all of the species which we can identify as sigma carbon-13, it is the community average value. And the community average carbon-13 value is higher in the reference site than the high CO2 site. So this data shows higher activity of the CCM in the present ocean. So the algae at the present CO2 level has been already enhanced, and they have already adapted for low CO2 levels. So they are not enhanced by the high CO2 condition. So based on the our results, there is some implication, and basically the ocean acidification, the effect of the ocean acidification has a negative impact on the ecosystem. For example, the loss of the biodiversity and the damage on the coral reef. So this is a negative impact for the human activity. And increase in the photosynthesis has been considered as positive aspect of the ocean acidification. So because if the photosynthesis is accelerated by the high CO2 concentration, it has a positive effect on the food supply or carbon sequestration, etc. But our result shows this positive effect will be limited. It's not so general case. So it's not so good the message for the prediction of the ocean acidification, but we should consider the effect which we cannot expect the CO2 fertilization. Okay, thank you for the listening. So our study is contributed by the various persons. So here the acknowledge. Thank you for everyone.
Thank you, Alessandra, for this very interesting talk. I think it highlights very much the strength of natural analog, as you can really consider the whole ecosystem rather than a single species. So we can take a few questions maybe. So reminder, you have a Q&A, and so you can type your questions, or you can raise your hand and we can give you, okay, someone already, James, okay, raise his hand. How can I give the talk, right to talk? Okay, people are raising their hand. James first.
Okay, can you hear me okay?
Yes.
Okay, thanks Shigeki for the great talk. It was really cool. This is a question actually I haven't asked you before, and it's kind of open-ended, but I found it really interesting how you used the typhoon to look at the different results. And I was wondering if down the line you had any plans for sort of taking into account increased incidence or strength of typhoons with global warming, because that could lead to some really, you could model different typhoon, business as usual as it is now, or more typhoons or less typhoons, and really see what happens to those ecosystems under these changing weather conditions as well. Is this something you've thought of, or is this something to work on in the future?
Yes, so the effect of the typhoon is the reported as my other colleagues, and also as you can see, as you said, so maybe the strength of the typhoon and the storm will be higher in future.
Yeah.
So that it's I think that not the impact will be the higher, but so in case of the Shikine, so in the future condition, in high CO2 area, before the typhoon season, the tough algae is dominated in the sea floor, and after the typhoon, it's completely flushed away. So it's a quite different situation in the present oceans. Present oceans, so the macroalgae is strongly attacked. But so in future, so in case of the Shikine, the tough algae is dominant by the diatom. So it will be attacked. So it's difficult to answer, but
it's difficult to answer, but I think to some of the forestry sort of papers where you have the rate of fire incidents and versus what become the dominant trees, for example, in a certain area, and you combine that with CO2, you could have some pretty interesting studies. But anyway, I hope your place is safe during the next typhoons next year. Cool talk. Thank you very much.
Thank you, James. Next, we have a question from Jason. Jason, can you talk?
Hello, everybody. Can you hear me?
Yes.
Yes.
Hi. I just wanted to say that I think that the chat is disabled. Well, it is for me anyway. So maybe Katerina can fix that.
You mean you cannot type your question?
Yeah, it says chat is disabled. So I wonder if some people can't ask questions because it's disabled.
You can put them in the
That's
queue.
just one thing. And the other thing, hello, Shigeki. Do they find the same things on land around CO2 seeps? What I mean is, on land, some botanists have studied the effect of CO2 on the plants. Does it have the same effect on land?
On land, so you mean not underwater?
No, I mean, they've studied CO2 seeps on land to look at the effects on the plants. I wonder if you see the similar effects, or don't you know?
You mean the homeland experiment? So in case of
yes, there's some studies looking at the botany on land.
Yeah, so you mean the area of the releasing the CO2 in the mountain, or the study on the land ecosystem, right?
Yes.
Yeah. Yes. I do not follow such kind of studies. So I heard there is a on the seat, and so maybe the productivity is enhanced. I heard the productivity is enhanced in the land ecosystem. Maybe so Florida. So the effect is completely different between the land and system and sea.
think you're right. I think on land, the productivity is definitely enhanced, but they don't have the time putting out the biomass every year. So that's the big difference.
Yeah. So one of the reasons, I think, this is my opinion. One of the reasons is the CCM. So because the algae develops the CCM, and so many algae parts this system. But I'm not sure about the land plants. But so it does not work so much. I think. Sorry, I'm not sure about that.
Okay, thank you.
And thank you. I think it's a good question. I think it's kind of assuming maybe the difference also here in your talk, you show that there is a change in community. And I don't know how much it's been considered for land, except for what James pointed out, the effect of fire. But it's true, it could be interesting to compare land and marine ecosystem. But again, it shows very much that the marine ecosystems are quite different. So we can have another question. Just a reminder, so for the questions, you need to type in the Q&A and not in the chat. So like you have a button in Zoom, like Q&A and not in the chat, and then we can get your question. Some more time. No one has any question. We'll keep some time at the end for additional questions. So if you're okay, I would like to go to the next speaker. Thanks again, Wada-san, for your presentation. And so our next speaker is Carlo Cattano from the Sicilian Marine Center, Stazione Zoologica Anton Dohrn in Italy. And so we'll shift from macroalgae to fish. And so Carlo has been studying mostly fish ecology and conservation. And he has studied the effects of OA on the physiological behavior and ecological response of marine fish through observation, but also experiments carried out in natural CO2 analogues. Now he's working also on the effects of different anthropogenic disturbance on fish community and trying to link this and improve conservation of threatened species and ecosystem. So without any more, give you the floor, Carlo. Can you start your presentation?
Thank you, Sylvain. Can you see the screen?
Yes, it's working very well.
Thank you. Thanks everyone for joining, and thanks Sylvain for the introduction. My name is Carlo Cattano. I'm a researcher at the Sicily Marine Center of the Stazione Zoologica Anton Dohrn in Italy. I have the great fortune to be part of the ICONA network, and today I will talk about some studies we carried out in different natural analogues to investigate how habitat changes that we observed under acidification condition may impact fish population and ecology and communities. So first of all, I'd like to start my talk to thank all the ICONA team. I had the fortune to work with some of them during these years, and I knew some other people during the last expedition in Palau. And these are the people who worked on most of the studies I will be presenting here today. So what we know about ocean acidification effects on marine organisms comes from more than 8,000 papers published since the early 2000s. And a considerable part of these studies dealt with the responses of fish under ocean acidification conditions. In general, we know that this large number of studies tells us that acidification may affect a wide range of species, for example, by changing gene expression, physiology, reproduction, and behavior. However, we can also say that different organisms may respond differently to acidification condition. For example, calcifying organisms may be affected and are generally considered more sensitive to high CO2 and low pH levels, whilst primary consumers, primary producers seems to benefit from increasing CO2. On the other hand, fish are generally considered more tolerant to acidification conditions, and this is why, because they have an efficient homeostatic system that allow them to maintain in balance their internal pH in high CO2 environments. However, reviews and meta-analysis found that some effects may be found also in fish, especially behavioral effects, and these effects are mostly linked to the alteration of functioning of GABA receptors. This review also found that the responses of fish are taxon-specific and trait-specific, with the early life stage generally more sensitive to high CO2 levels, and also found that the magnitude of the impact changes according to PCO2 levels. We have also to consider that most of our knowledge comes from experiments carried out on single species and in short-term lab-controlled conditions, and very few of these studies considered species interactions, and so for this reason it's difficult to study organisms in their ecological context and to scale up from organism responses to community and ecosystem level effect. So to address this issue, natural analogues of ocean acidification have been increasingly used in different parts of the world. In this map, you can see the distribution of CO2 seeps and CO2 vents in the world and in temperate, subtropical, and tropical area of the oceans. And natural analogues are systems where environment characteristics are similar to those expected to occur in the future due to CO2 emissions in the atmosphere. And this system might be used to study the effects of acidification on marine life by observing species in their natural environment and considering also the community level responses. The ICONA project aims to facilitate innovative research on the ecosystem level effects of ocean acidification using natural analogues, and another aim is to develop guidelines for research in these systems. In the map, you can see the main localities, the main areas where the ICONA team is working, and in particular in Vulcano, Panarea, and Ischia Islands in Italy, in Shikine Island in Japan, in Nico Bay in Palau, in Papua New Guinea, and in New Caledonia. So some of these areas are CO2 seeps, some other are semi-enclosed bay. And in general, we know from the literature that the studies carried out in these and other CO2 seeps, especially from tropical and subtropical areas, show that acidification changes the composition and the structure of the biogenic habitats. And this change may lead to less diverse and complex habitats, with this potentially affecting also the ecosystems and the service they provide. But what is still unclear is how do fish respond to these changes in the structure and composition of habitats. We carried out studies in different natural analogues to understand how fish respond to these alterations of the habitat, and what can be the impact at population and community levels. Today, I will present some of these studies that have been carried out along natural CO2 gradients in Italy and Japan, in particular in Vulcano Island and in Shigeki Island. So Vulcano Island is host an extensively studied CO2 seep in the Baia di Levante, where CO2 emission, mostly CO2 emissions are present and generate a CO2 gradient that runs parallel to the coast in shallow water. Many experiments have been carried out since 2008 in this area, and in particular in considering two main CO2 stations. One station is characterized by CO2 level expected to occur at the end of this century, so around 800-1000 PCO2 microatmosphere, and the other one is on the eastern part of the bay and is characterized by current day CO2 levels. The comparison of these stations show a quite clear habitat shift from habitat mostly dominated by macroalgae and coralline algae to an habitat dominated by turf, mostly by turf. We also know that the PCO2 values are in these stations are not constant, but show daily variations that are mainly due to biological activity of the communities, and these variations are more evident in the high CO2 locations, where CO2 concentrations range from 400 to more than 1000 PCO2 microatmosphere on a daily basis. In these stations, we performed this experiment to understand how the habitat changed the performance of the species, and we mostly used two model species that are Symphodus ocellatus and Gobius synconitus. These species were chosen because they are very abundant in both the locations and the CO2 stations, and also because they have a narrow home range and are closely show a close association with some habitats. In particular, Symphodus ocellatus show a very strict association with benthic habitat, especially during the reproductive season, and Gobius synconitus associate with anemone for sheltering. So Symphodus ocellatus is a nesting labrid very common in the rocky reef of the Mediterranean Sea. This species has a very complex reproductive behavior that involve a different typology of reproductive males, and the nesting males that is bigger than the other males build nest with algae. The male attract the female to this nest, where the female lay the eggs that are immediately suddenly fertilized. We also know that in normal CO2 conditions, this nest, the nesting male shows display a preference for the algae that are used for the nest. In particular, the nesting male actively select the coralline algae, probably because they may confer resistance to the nest and may enhance the structure of the nest. So Iania rubens is the main, as you can see, the main algae found in the nest, but at high CO2 conditions, under high CO2 conditions, these algae disappear. So the composition of the nest change, and our question is how ocean acidification conditions may affect the development of the eggs inside the nest. We then performed a translocation experiment to understand if elevated CO2 change the metabolic rate of and so the oxygen consumption of the eggs, and then if these conditions may affect also the larvae when they hatch. We collected portion of nest from with embryos from high and ambient CO2 conditions and reciprocally transplanted them for 30 hours. And we found that oxygen consumption did not change between CO2 sites, but embryos collected from ambient condition and translocated to high CO2 conditions showed higher metabolic rates and led to smaller larvae. Also we can these results suggest that offspring from the high CO2 nesting site could be resilient to a wider range of PCO2 levels than the offspring coming from the present-day PCO2 levels. In the frame of the ICONA project, we recently replicated this experiment, this translocation experiment, to study the transcriptomic of the eggs and to better understand what are the molecular mechanisms that may explain this tolerance shown by eggs to acidification conditions. The other experiment I will describe was led by Davide Spatafora and was recently published. The study focused on Gobis incognitus, a cryptobenthic species that uses the sea anemone, Anemonia viridis, for sheltering. We observed that under CO2 conditions, both the sea anemones and the gobbies were more abundant, and that the association rate between the gobbi and the anemones did not change between CO2 sites. We then ran an in situ experiment with a cage to investigate potential behavioral alteration under CO2 acidification condition. And we translocated, also in this case, we translocated adult gobbies from ambient to high CO2 conditions and vice versa to record their association with the anemone and to record their behavior in presence of a predatory threat. The experiments didn't show major behavioral disruption of the gobbi under elevated CO2 regarding the use of the shelter. However, fish from ambient conditions showed unexpected reduction in the swimming activity after the presentation of the predatory threats, while such changes were not detected in the other CO2 treatments. This suggests an effect of CO2 on the gobbi anti-predator strategy. But the higher population density of gobbies at the CO2 seeps combined with the limited behavioral effect that we recorded in this experiment may suggest that these fish living at high CO2 conditions show behavioral plasticity and also an adaptive potential to high CO2 conditions and could benefit from increased habitat availability. Very recently, a paper published in 2023 showed the molecular mechanism underpinning the tolerance of anemone gobbi to acidification condition. And some members of the ICONA members were involved in this study, and they found that there were differences in the expression of transcripts involved in pH homeostasis, cellular stress, metabolism, and other processes that may facilitate acclimation and tolerance. So moving from population to community level effects, here I will talk about a study we carried out in Shigeki Island, in the Izu Island in Japan. As Wada-san said before, these CO2 seeps are located in a temperate subtropical transition zone. The emission area has been extensively described in recent years, and two main field stations have been used to understand the effect of acidification on benthic communities. One of these stations is the control site with the current day PCO2 levels, and the other one is where the CO2 emissions are present is characterized by a gradient, a PCO2 gradients ranging with PCO2 values ranging from 400 to more than 1,000, 2,000 microatmospheres. So Agostini and colleagues in 2018 described a transition from highly diverse habitats dominated by complex corals and macroalgae to simplified habitats mostly dominated by caulerpa and turf. And these transitions led to a reduction in the complexity of the habitats. And this reduction started at 400 at moderate CO2 levels. We carried out this study in these two main CO2 locations in Shigeki Island to understand if this habitat shift could lead to difference in the composition and the structure of fish communities. To do that, we used different sampling methods, including photo quadrats and underwater visual census to characterize the habitat and the fish composition, the fish community composition. And of course, these methods were replicated along the CO2 gradient and in the different CO2 locations. So the results confirmed what Agostini and colleagues found a couple of years before regarding the habitat composition and the shift along the CO2 gradient. We found that table and encrusting corals were common at control sites, but disappeared at elevated CO2 sites in both sampling periods, because we, as you can see in the figure, we replicated this survey during 2 seasons, before and after the typhoon season. In June, we found in the first sampling season, we found dense mats of caulerpa and turf dominated most of the habitat at elevated CO2 sites. In September, most of these mats disappeared due to the typhoon activities, and coralline algae and low-profile turf were dominant. We also found that this habitat shift caused a decrease in the complexity, of habitat, and this decrease in habitat complexity reached the minimum value at moderate CO2 levels and increased again at higher CO2 conditions, but without reaching the same complexity we found in the ambient site. So here we can see what we have seen along the PCO2 gradient. In general, we found a very complex fish community associated with a very complex habitat community, biogenic habitat community. Along the PCO2 gradient, we have seen that this diversity decreased, the diversity of fish decreased, of species of richness, and reaching the minimum value at very high CO2 levels. As expected, we found that this decreased richness of fish species with increasing CO2. And this change was best explained by 5 benthic variables: the cover of table and massive corals, non-canopy algae, turf, and CCA. The difference in the composition and structure of fish assemblage were mostly due to a decrease in tropical, subtropical, and coral-associated species at elevated CO2. Also, we found a higher abundance of herbivores at elevated CO2 conditions, whilst the abundance of planktivorous and carnivorous decreased at high CO2 levels. So this suggests that habitat shift may lead to fish community reshuffling, probably driven by the loss of resources for fish, for example, food and space, and that such changes in fish communities may cause major changes in trophic processes and energy pathways, thus potentially affecting ecosystem services. In the frame of the ICONA network, we are replicating this experiment and these observations in other tropical CO2 seeps and semi-enclosed bays by using the same methods, the same sampling methods, and also other sampling methods. For example, we are using BRAVs, Baited Remote Underwater Video, to sample fish fauna and in particular predators and high trophy level species. These observations are mostly carried out in Italy, in Vulcano and Panarea, but also in Palau, in Papua New Guinea, and in other CO2 seeps. For example, in the same protocol was that we used in Japan has been recently applied in Nico Bay, a semi-enclosed bay in Palau. We were there last spring, and much work was done to characterize fish communities under different CO2 conditions. The elevated PCO2 levels observed in this bay are mainly due to natural biological activity and seawater circulation. Nico Bay is also characterized by multi-stressor conditions, particularly for CO2 and temperature. And this makes this bay a unique site for evaluating the responses of organisms to warmed and acidified conditions. A recent work by Kuriara and colleagues showed that hard coral coverage does not change with increasing CO2 and temperature, but the diversity and the community composition do change. In this context, we replicated the protocol to describe fish community, and also performed an in situ experiment to study the behavior of the pajama cardinal fish that is quite common in the area, and how the association of this fish with the two species of corals may change under different CO2 conditions. So in conclusion, we can say that acidification will probably simplify habitats and will change the associated communities and will lead to a decreased fish diversity. Our studies add to previous evidence that acidification will change the ecosystem, but some species seem to be able to cope with expected acidification conditions, and they display behavioral plasticity and adaptive molecular mechanism. Natural acidification analogues may be used in combination with experiment in the lab to study how habitat shift observed may change species performance. At the same time, this system provides a unique opportunity to understand acclimatization and adaptive mechanism of species to these conditions. And the ICONA network is making possible the application of a multidisciplinary approach in different systems around the world, thus providing opportunities to better understand how global change will affect marine population and communities. Thank you.
Thank you, Carlo, for this talk. Very interesting. Once again, some studies that can only be done in special sites, I would say. So we have maybe a little bit of time for a couple of questions. If anyone has questions, you can write it in the Q&A box or raise by raising your hand. In the meantime, I have a question for you, Carlo, actually. You say that the community shift, but did you ever try to assess the biomass of edible fish? Because if you are thinking in terms of ecosystem service, how much protein we can get from this future ecosystem, you know?
Thank you, Sylvain. No, we don't have an estimation of biomass, of fish biomass under two conditions. We know the abundance of fish. Of course, this is a very interesting topic, but for considering the methods we are using, we need to use very specific systems to estimate the biomass of fish. So, of course, this can be a future step, a next step in this research.
Okay, thank you. Okay, we'll have some time maybe at the end of this session. Okay, there is a question now. There is a question from someone anonymous, so it will be different. And but the question is the study still in progress for Papua New Guinea? So did you run a similar study in Papua New Guinea? The question.
So I didn't go there, but the fish team of the ICONA network is working there and collected collected data on fish community in the different conditions. We know that in Papua New Guinea there is a transition from hard coral to mostly massive corals, and this change in the habitat composition we think may lead to change also on fish community, especially for those species that live closely associated with branching corals, for example, but also for other species. As we have seen in Japan, what we found was that not only these species strictly associated with the bottom changed, but also other species changed, probably because not only the diversity and the change, but also the interactions between species may lead to different communities. So we replicated we replicated the protocol also in Papua New Guinea, and the data we are analyzing still the data.
Okay, so it's on the way. Thank you. We're running a little out of time, so I would like to go to our next speaker, which will be Ayaka. Thank you, Carlo.
Thank you, Sylvain.
You have to stop sharing.
Yeah, I don't know how.
Up. Stop share.
Okay.
I did it. Okay, so our next speaker is a young, promising scientist, Ayaka Umeda. She's a first-year master at the University of Ryukyu in Okinawa, Japan. And she's been looking at the benthic community in natural analogues environment, especially like the low pH coral reef ecosystem. And she aims to utilize the survey to include the understudied benthic organisms and understand the potential of these groups for the to dominate and persist in the future environment, especially under oceanification. So Ayaka, can you start your presentation, your camera and presentation too? We can't hear you, so maybe you're muted.
Can you hear me?
Yes, we can hear you.
Okay, you can see everything?
We can see your presentation, yes.
Perfect. Well, thank you so much for the introduction, Sylvain. So today I'll be presenting some of my preliminary data on the benthos, where we can peek into some of the benthic groups that are dominating Palau's natural analog environments. So anthropogenic driven effects of climate change have and will continue to change our ocean's chemistry. The increase of CO2 emitted into the atmosphere is causing ocean acidification, as our oceans act as a major carbon sink. And the consequences of this could include the negative impacts on functional processes in marine organisms, as well as the reduction of calcium carbonate mineral saturation, and that can pose a threat to marine calcifiers that need to build skeletons and shells. And the effects of ocean acidification on coral reefs are still being studied. However, current research suggests that as the pH decreases, there could be a shift from calcareous to non-calcareous communities, some of which could be dominated by algae. And generally, coral diversity will likely decrease, even if the abundance of hard corals isn't affected at some sites. And there have been many lab studies done on ocean acidification in controlled environments, However, natural analogues, which can emulate future projections of pH and temperature, are increasingly being investigated to see the potential impacts in natural systems with multiple influencing factors. And coral reefs are a larger concern because there are many organisms that depend on the structures of some corals. And most of the studies today focus on organisms such as hard corals, which are important reef-building organisms. But there are also understudied organisms or more understudied organisms such as sponges, which are filter feeders, soft corals, which are competitive benthic organisms, which can be more structurally tolerant to ocean acidification, and zoantherians, which also have traits that make them structurally more resilient competitors. And all of these traits allow them to persist under environmental pressures, which is why they also need to be properly investigated to get a clearer picture of how our marine ecosystems might change with ocean acidification in the future. So in order to kind of create a holistic look at benthic groups in Palau, I'm asking the question, what are the patterns in benthic community structures between low pH and control sites? And in order to answer this question, I'm first looking at Palau as the first geographical area. And Palau is an island nation and an archipelago of 340 coral and volcanic islands, and it's located in the Western Pacific Ocean over here. And Palau has a natural analog site called Nico Bay, which is a semi-enclosed bay with long residence times and can lead to warmer temperatures and lower pH compared to the surrounding reefs. Previous studies, such as Kurihara, this one done by Dr. Kurihara, has measured the pH and studied various sites around Nico Bay to look at marine organisms that live in these environments, such as hard corals. And for the purposes of my presentation today, I've selected 3 inner bay sites, Nico 5, inner site 29, and Nico 3, which are all have varying pH from low, medium to higher pH. And I also selected 3 control sites, the super duper control, fish control, and the lighthouse. The pH from the lighthouse is still in progress due to issues with the probe, but as an outer bay control site, the pH range should still be around 8, a little over. In terms of the methods, 3 10-meter transects relate at 2 depths, and for this presentation, I'm only looking at 1 depth at 3 to 5 meters for consistency. And photos were taken 1 meter above the transects, and I used the photo intercept transect methods to analyze the photos. And through the photo analysis, I calculated the percent cover. And for now, I've only identified the general benthic components and will later analyze them to a more finer and detailed taxonomic level in the future. I then used the percent cover and did some further multivariate analyses, which we will go into. In terms of my results and discussion. So I first looked at whether we could see a large and general difference in benthic composition between the inner bay, which is seen here in yellow, and the outer bay, which is seen in blue. And here in this graph, we can see that hard corals are dominant compared to other benthic groups in both the inner bay and the control sites. And it also shows that sponges are prevalent in the inner sites compared to the control sites. However, in terms of benthic community structure, the ANOVA analyses and the analysis of similarity showed that the differences are not actually significant, statistically significant. And even though the differences are insignificant, looking at the SIMPER results, it's interesting to note that the components that showed the most dissimilarity between the inner and outer sites is the sponges, which is seen in blue here. And it showed the largest percent contribution of dissimilarity of 13.96%. And here, when we looked at the differences in terms of community assemblages between the sites, and these are the 13 categories that my analysis was broken down into, we found that this actually was statistically significant. And in order to better visualize the information comparing the benthic categories between sites, we can look at this cluster analysis, which shows that while we would expect all of the inner bay and the control sites to be similar to each other, NICO3 and the super duper control were actually a little more similar to each other compared to the rest. And this could be because of the similarities in the pH and also maybe the environment that they're in. So now that we kind of understand the sites a little better, we can come back and look at some of the notable points from this graph. Firstly, we can see that there's a dominance of hard corals in all the sites inside Nico Bay and even in the controls. And we can see the relative percent cover of the benthic categories within each site here. And when running the similarity of percentages test, we can see that hard corals, which is labeled as the orange portion of the graph, largely characterizes each site with percent contributions that range from 21% to 53%. And we can see that in general, there are more hard corals in the sites inside the bay compared to outside the control sites. And those results may seem a bit counterintuitive because hard corals are doing well in sites that are more acidified. But as it's kind of been touched on, some studies have looked at this pattern, and one of the hypotheses is the ability for these corals to acclimatize. One study found low bleaching mortality inside the bay compared to other sites, which can be seen in this figure on the top left, and this might be due to acclimatization because of near constant exposure to high temperatures, which can be seen in the figure on the right. And another study also hypothesized that corals might be acclimatized to low pH conditions because of self-seeding and isolation due to the fact that Nico Bay is enclosed, and they modeled the long residence time of approximately 71 days, measured by tracking and releasing virtual waterborne particles. And because of this, there would be a lot less mixing with genetic material found outside the bay and allow for selective pressure for traits that would allow corals to survive under environmental conditions found within the bay. And another interesting point is focusing on the sponges and algae. So here we can see that sponges are prevalent in the transects inside the bay, which can be seen as the yellow, orange, and red bars here, but not outside the bay, which wasn't actually detected in the transect pictures. And we can also see that comparing Intersite 29 and Nico 5 and looking at the same sites with the algae, there is an inverse pattern, which is pretty interesting to note. And so here we can see the relative percent cover of benthic categories within each site again, with sponges representing the blue and algae representing the mint green. Sponges are one of the larger biotic components that characterizes the inner sites, and they aren't found at all in the control sites. And here we can also more visually see the inverse pattern between algae and sponges, which is reflected also in the relative percent and similarity of percentages analyses. And we can also note that with that inverse pattern, the control sites also have more algae than inside the bay. And so what might help explain the abundance of sponges in the inner bay is firstly, potentially the water quality in the area. These surveys aren't conducted in a controlled environment, and so pH isn't the only factor that could impact the benthic communities here. And studies have shown that water with lower quality, that has higher nitrogen levels and pollutants can actually help the sponges grow faster. And another factor that would impact the abundance of sponges is the presence of sediment, which kind of falls under the water quality point. And while some sponges may be able to adapt or have some tolerance, a lot of sponges experience physiological stress in areas with high sedimentation because it can clog or interfere with its pumping apparatus and its ability to filter feed. And finally, the inverse kind of pattern we see between sponge coverage and algae is also notable. In previous studies, an inverse pattern between turf algae and sponges has been looked at before. And for further analysis, I plan to separate groups of algae, so coralline algae and turf algae, etc. But through the analysis of the images so far, I've definitely noticed the presence of turf algae, which could help explain this pattern if there is a correlation between these two groups. And this could be because of competition for space, as turf algae is also a very strong benthic competitor. And when examining pictures and looking at the reef while doing the surveys, we noticed large outbreaks of one particular sponge, which is shown in the images on the right. And due to its abundance, it would be important to try and identify and understand what impact it might be having on the reef. And although my data can't make any conclusions on the role or impact this sponge has on the reef, I'm currently thinking about two potential hypotheses. The first is that this could be another coral killing sponge. As of right now, there are a few known coral killing sponges, including Terpios, Calinula, and Clathria. And in order to help identify the sponge, further molecular analyses and morphological analyses would be needed. And beyond this, further studies and monitoring will probably be required to look to see whether these sponges are actually encrusting and killing live corals over time and creating these potential outbreaks in the Palau Reef. And another idea is that it's a dominant bioeroding sponge. And according to some of the studies, sponge biomass and bioerosion rates are predicted to increase under the effects of ocean acidification. And rapid bioerosion, which breaks down the calcium carbonate, can have a lot of negative consequences on both the topography of the reef, as well as on other hard corals and macroorganisms, because it impacts the ability of these organisms to maintain calcium carbonate skeletons. And additionally, when bioeroding sponges grow close to or in the skeleton of organisms, they can act as parasites, draining the energy of the host, and that can lead to the death of the host. And so the ability of these sponges to persist in these environments could allow them to potentially dominate the reefs in the future. And so it would be important to take a note of this for future studies. And so some key takeaways from my preliminary data so far is that reefs in Palau are dominated by hard corals. Sponges are more abundant in Eco Bay compared to control sites, and the sponges might be more adapted to conditions in Eco Bay. And a couple of my future steps is further analysis of my transect data. Again, this is like a very small piece of all of the data that I still have to go through. And I really want to increase the taxonomic resolution to which I'm analyzing it with. And I also want to supplement this data with eDNA. And furthermore, I would like to try and do some molecular analyses of collected samples of the unidentified sponge. And it would be great to also visit a few more sites to try and compare different trends regarding the benthic communities there. Thank you so much. And thank you to everyone else who helped.
Thank you, Ayaka. Yeah, I think you'll have the occasion through the ICONA network to visit other sites. I like your super duper control. So you kept the name. And so we are quite late in time, but maybe one question. I see that someone is raising their hand. We take only this question. Jason.
Hello, Ayaka. It looks like a fantastic place. I was thinking during your talk that perhaps it wasn't a very good analog for the future because of the nutrients, etc. But then I changed my mind because, of course, we're causing eutrophication. We're causing stress to coral reefs all over the place. And perhaps it is a very good analog. So what do you think?
I think it would also be a pretty good analog because again, it's not just all of the pH and the temperature and everything, the isolated changing water chemistry there that's affecting the benthic communities. It's also like sedimentation and runoff and all of those things that are also, well, they're all anthropogenic, but also directly inputted by humans. So in that sense, I think it is also a pretty important site to keep in mind.
Thank you. I agree.
Yeah, I agree too. I think multi-stress is maybe what will define the future oceans, and we see that maybe we'll have to talk about sponge reef instead of coral reef, like we've seen in the Caribbean, actually. Okay, thank you, Ayaka, for the talk. I would like to go to our next speakers, Leila Ijima-san, who is also from the University of Tsukuba. And Leila is studying the coral ecophysiology at the Tsukuba, and she hopes, but I think her research can contribute scientific knowledge. She's also a first-year master, so again, early career. And Leila, if you can start your talk, so we don't need time.
Can you see my slide now?
Yeah, no problem. We can hear you and see.
Okay. Okay. So thank you for the introduction. Today I'm going to talk about how hermatypic corals change their physiological response with ocean acidification under the reduced light availability. So as you might be familiar with, but ocean acidification is a phenomenon caused by increased anthropogenic carbon dioxide emission into the ocean, which leads to lower oceanic pH and carbonate ion abundance. And this is expected to impact the diversity and abundance of the calcifying organisms, which forms calcium carbonate skeleton, like hermatypic corals. And hermatypic corals are a colonial cnidarian, and they are symbiotic with photosynthetic dinoflagellates symbiodiniaceae, which is generally called zooxanthellae. And when the zooxanthellae photosynthesize, they translocate about 95% of their photosynthetic product to the coral host. And then coral hosts convert this energy into ATP at their mitochondria and use it to maintain their metabolism and protein lipid biosynthesis and to produce their calcium carbonate skeleton. However, under acidified condition, corals will need to spend a large amount of energy to emit the excess hydrogen ion inside their tissue and also to prevent their skeleton dissolution. So this increase in energetic demands of corals under ocean acidification, it can also reduces the coral host metabolism and their biosynthesis rates. And in temperate areas like Japan, these areas are expected to be a future coral refugia for some tropical and subtropical corals that are expected to expand forward due to ocean warming. However, the effect of ocean acidification is stronger in the higher latitudes compared to the temperate, it's stronger in the temperate regions compared to the tropical regions. So the corals in the temperate regions, they will be sandwiched between the low pH region and the high temperature regions. And moreover, there are less light available for zooxanthellae to photosynthesize. So these factors might strongly affect the coral physiology, which might eventually limit the coral distribution in depth and in latitude. And talking about the light availability, it is known that lower light intensities decreases the coral metabolism and the calcification. And as ocean acidification causes greater coral energetic requirements, corals under combined ocean acidification and low light will face an unbalanced energy requirements and inputs. So we hypothesize that corals under low light condition would exhibit a stronger effect of OA. And to test our hypothesis, we conducted 6 months transplantation experiment and 1 month incubation experiment using the warm temperate coral Acropora soltariensis, which is known that forward expanding species. And we conducted the coral sampling and transplantation at Shigeki Island in Japan, where it has a volcanic CO2 seep that bubbles at both shallow and deep depths. And we transplanted the coral microcolonies at each present-day pH site and OA site in both 5-meter and 15-meter depths for 6 months in the field. And later, we conducted 1 month incubation experiment using the same condition. Then after each experiment was finished, we measured photosynthetic efficiency and photosynthesis rate as the symbiont activity, and respiration rate and protein concentration as coral host activity, and the calcification rates as the calcium carbonate production. So for the results, in this page, the results of FVFM, which is the maximum photosystem to photosynthetic efficiency, is shown on the left, and the gross photosynthesis rate is on the right. And inside the figure, left result shows the highlight treatment, and low light is on the right side. And the top results are field experiment, and the lab results are on the bottom. And the orange color represents the acidified site. So for the results of the FVFM, it showed an increase under low light in the field experiment, and moreover, it showed an increase with acidified treatments in both field and lab experiment. And usually, higher FVFM rates means that it has the potential to efficiently activate their photosystem even under reduced light availability. But the actual photosynthesis rate by the zooxanthellae decreased under low light, which might mean that the positive effect of OA on FVFM was not enough to compensate the reduced light availability to increase their photosynthesis rates. And when we focus on the lab experiment, we can see that the effect of OA under low light was slightly mitigated compared to OA highlight treatment. And this slight increase of photosynthesis under OA low light treatment might reflect the positive effect of ocean acidification on the FVFM rate. And when we look at the coral respiration, which is a metabolic process by the coral host, it decreased under low light in both experiments, which might be due to the reduced autotrophic input by the zooxanthellae. And for the protein concentration, which is their metabolite, it also decreased under low light in the field experiment. And again, when we focus to the lab experiment, the small increase of the photosynthesis that is deriving from the increased photosynthetic efficiency is probably affecting the little increase of both dark respiration rate and the protein concentration. However, corals in the field experiment did not change their metabolic rates due to ocean acidification. And this different response of OA on between the 6-month field experiment and 1 month lab experiment might be due to the difference of the acute and chronic response of ocean acidification. And when we look at their calcification rates, consistent with their upper stream activities, light calcification rate reduced under low light. But again, in the lab experiment, the acute response of ocean acidification under the low light was also observed in this light calcification too. And for the dark calcification rate, although lab corals showed a decrease under low light and did not show effect of OA, field dark calcification rate showed a decrease under ocean acidification, and also the dark calcification rate showed a larger decrease under low light with OA, although non-significant. And however, the dark calcification rate decreased with OA in the field, it did not affect on their actual skeletal growth within the experimental period, and their accretion rate showed a decrease with low light. And for the lab corals, we couldn't see the positive effect of OA in either their calcification rate or their accretion rate. And overall, we could see the stronger effect of OA only in the long CO2 exposure. And when we focus on the effect of ocean acidification, in the lab experiment, we could see a positive effect of increased FVFM under OA leading to a little input of the photosynthetic products, which might also lead it to the increase on their downstream activities under low light. But such positive effect of OA was not observed in the 6-month field experiment. And this suggests that the effect of OA might be more prominent in the further long experimental duration. But overall, the coral we used, Acroporus soltariensis, did not get a combined effect of OA and low light within the 6-month experimental period. And this potential acclimatization capacity might be the reason why these species are capable of poleward expansion. However, the abundance of Acroporus soltariensis in the Shigeki Island CO2 seep is quite low. So this might mean that this species might get an impact of ocean acidification during their lifetime CO2 exposure. And the response of corals on acidification and different light availability is known to be species specific. So in order to investigate if other species, if other coral zooxanthellae holobionts would show an unbalanced energy requirement and inputs during their lifetime exposure, we conducted a similar experiment, but this time a cross-transplantation experiment using the natural analog in the Republic of Palau, which is the Nico Bay. And Nico Bay is different from the CO2 seep in the Shigeki, is that it is a semi-enclosed bay and has a low pH and higher increased temperature due to the low water circulation within the bay. And we used the coral Poecilopora acuta and Porites rus, originating both from the present-day pH site and the Nico Bay, which is the acidified site, and cross-transplanted it for 5 months this year. And this is preliminary data of the Palau experiment. And the figure on the left showed the experiment conducted in Japan, and the middle figure is the results of Poecilopora acuta, and the right figure is Porites rus. And one thing is that Poecilopora acuta, originating from the control site, which is transplanted to OA highlight site, showed a complete mortality. But other than that, all corals showed an increase of FVFM under acidified treatment, including the experiment conducted in Japan. And these results suggest that zooxanthellae might have the ability to benefit from the increased carbon dioxide concentration, which might allow them to efficiently activate their photosystem. However, the increase of FVFM did not also affect their actual skeletal growth, and it showed a decrease under low light in all experiments, also including the experiment conducted in Japan. And in addition, Porites rus, originating from the Nico Bay, showed a higher accretion rate compared to the Porites rus originating from the control site. And this might suggest that that Porites rus have the potential adaptation to the ocean acidification. So to conclude, in our study, the main factor that affected the coral physiology was the reduced light availability, and the effect of ocean acidification was rarely seen. And contrary to our expectation, there were no combined effects of OA under reduced light availability. So these results might suggest that the warm temperate coral Acroporus soltariensis, their future distribution in the temperate area might not be limited in either in depth and in latitude. However, longer CO2 exposure would be crucial to investigate how the coral and symbiont holobiont would change their physiological response under the chronic stress of OA. But overall, our studies suggest that some coral have the potential acclimatization and adaptation capacity to the change in the oceanic pH environment. that's all for my presentation. Thank you.
Thank you, Leila, for the presentation. So very much running late. So I would like to ask the audience if you have questions regarding Leila or anyone, any other speaker. We can take a few questions, maybe. Questions? Yes. Oh, we have questions. So who was the first? Like James,
Yeah.
you can talk.
Okay, cool. Thanks, Leila, for the talk. It was really cool. It was cool to see how you're putting things together. I had a simple question, and it's something that I've sort of been thinking about for some time. So there's not a lot published yet, but recently many people think, well, some people think that some of the corals that you find up in Shigeki are actually native species that aren't moving north, but have always been there. And that doesn't take anything away from your talk, because I think, you know, the corals are going to be faced with colder temperatures to the north and increasing temperatures from the south. But if the corals at Shigeki were native corals that are experiencing climate change, would it change any of your conclusions or your big take-home messages or not? I'm just sort of curious what you think.
Thank you for your question. If the corals we used in this experiment was native in Shigeki.
I mean, native as in not expanding north, but have, you know, like have always been there, kind of.
Yeah, I think that's a good question. I think we need to look at their genotype and to see if they're native or not. But one thing I think it's important is that they showed an maybe I can say acclimatization to that some kind of very stressful environment.
Yeah.
And maybe those species might be stressed in the future, maybe end of the century, and they might go further. The native species in the Shigeki might go further. So I think maybe we can assess that if they can go further or not, if they are further north or not. Sorry.
Yeah, no, that makes sense. I also think, you know, like what you showed with the acclimatization really sort of reflects also what was seen in Palau, not only with pH, but with, you know, higher nutrient levels and stuff. So, you know, it's not black and white. And I think with multiple stressors and multiple things going on, it's really hard in these natural analogues to tease them out. But I think it actually makes them more important sometimes than some of the controlled experiments. So keep going. Best of luck. Good luck.
Thank you so much.
Thank you, James. So we are very much running out of time, but the team raised his hand. Maybe, Tim, if you have a short question. Tim, you should be up if you unmute now.
No, I don't have any question.
Oh, okay, you're
sorry.
Okay.
It was a mistake. But you know, I just want to say great talk, everyone, and I really enjoyed. So thank you.
Thank you very much.
And thank you.
Thank you. No problem. Thank you to all speakers. And well, it was a great session. If you want to know more, but Catalina shared the link about the ICONA network, please check our website and you can contact us and you can see if you want to use this kind of very interesting site. Thanks again for GOA-ON and the different facilitator, our host. And I would like to give back the talk, the mic to the Secretariat of GOA-ON. Thank you very much.
Thank you so much, everybody, for these really fascinating presentations. So we would like to thank the organizers, namely Sylvain, and all the presenters for participating in Ocean Acidification Week. This was the first session of OA Week 2023, and I think it was a great success. So thank you very much. Thank you for the audience for attending, for asking all your questions. Please don't forget to consult the GOA-ON website for the schedule of any upcoming presentations. We have 3 more to go today alone. And if you would like to stay up to date with the GOA-ON community, please consider signing up as a GOA-ON member on the GOA-ON website. And to discover more about ocean acidification research for sustainability, the OA UN Ocean Decade Program, please scan this QR code and it'll take you straight to the sites of our latest venture. So with that, thank you very much, and we will see you again next time. Goodbye.